Hello. So I'm one of the lucky guys who had early access to this exciting platform, and I really want to congratulate Mission Bio of developing this very exciting single-cell sequencing platform. And today I would like to share our early experiences on using this platform on the real AML patients' bone marrow samples. So I have no financial disclosure to declare. So Dennis already went over some of the aspects why this platform is suitable for AML. But AML, almost 60-70% of patients can achieve what we call complete remission. However, half of them eventually relapse. And as a clinician who treats this AML patients, this is really the pressing needs to solve, because after AML relapse, it's much more harder to put back them into another remission, and the outcome after the relapse is extremely poor. And with the advent of this next-gen sequencing in the past decade or so, we have learned so much that clonal heterogeneity and the clonal evolution is associated with this relapse. As shown in this right lower figure in the Nature paper, that certain subclone expands at the time of relapse, which may be accountable for this relapse mechanism. Unfortunately, as Charlie mentioned, that there is a fundamental limitation of inferring the clonal architecture based on the bulk sequencing that can be confounded by tumor purity and also the zygosity of the mutations. And also, bulk sequencing can never really able to tell difference between the independent clone and also the subclone of the bigger founding clones. With this background, we thought that the Tapestri platform could answer some of the answers and also questions, and also overcome some of these hurdles that in the bulk sequencing. So as a pilot study, we studied about 3 patients of AML who received chemotherapy in our department and achieved complete remission and subsequently relapsed. And we captured different bone marrow samples from the different time points, simply baseline and also at the time of remission and also at the time of relapse. Unfortunately, for the 2 patients, we were not able to capture the remission samples. So for the 1 patient, we had a 3 time points analysis, and the other 2 patients, we had a 2 time points analysis. And we run this Tapestri platform on these longitudinal bone marrow sample that was acquired. I'm going to go through these 3 cases. And the first case is a very typical AML patient who is a 66-year-old man who are diagnosed with AML, what we call FAB classification M5, and achieved complete remission after induction chemotherapy. But unfortunately, after 3 months later, he experienced the relapse. And through the clinical bulk sequencing, we already knew that this patient had these 3 oncogenic mutations: TP53, DNMT3A, and also FLT3-ITD. And as you can see in the chart there, there's a longitudinal difference of variant allele frequency that was estimated by this bulk sequencing. So we run these exactly the same time point samples with this Tapestri platform. This is a QC metrics from this first patient run. On all 3 time points, in average, about 5,000 cells were able to be genotyped. And there was a spike in the Raju cell, which kind of served as an estimated allele dropout rate. And we kind of estimated the dropout rate anywhere from 2.8% to 8.4%. This Tapestri platform detected total 17 variants, of which 4 of them were non-synonymous variants, and 3 of them were exactly same variant that was detected from the bulk sequencing. And this variant calling was performed in a blinded fashion that the bioinformatics who called these variants did not know what mutation was detected from the bulk sequencing. And the one variant that was not in the bulk sequencing, which is an ASXL1 variant, that turned out to be actually the whole, germline polymorphisms. As you can see here in the left, that ASX1 variant has almost 100% allele frequency in all cells. So it also supports that these are the germline polymorphisms. And the 3 variants that were detected by the bulk sequencing, this Tapestri was also able to detect. And as you can see in the middle, there's a striking clonal heterogeneity that Tapestri suggested. Obviously, there is a big chunk of cells which carry all these three mutations, which most likely is the founder clone. But in addition to that, there are multiple patterns of subclones which carried combination of certain mutation. And some of them, the Tapestri suggested that the mutations were homozygous, especially in this DNMT3A. In the center is a remission sample, and it's very hard to see that there was an variant detected, but actually there were total 11 cells out of about 5,000 that carried abnormal cells, which most likely represent this minimum residual disease. Another surprising finding was that the diagnosis and relapse sample really had very similar clonal architecture. It's almost like every clone was preserved from the diagnosis to the relapse. And the left side of the bar graph is a comparison of the variant allele frequency that was estimated by the bulk sequencing versus this single cell platform. And you can see that there's a very good concordance of VAF estimate from the bulk and also the single cell sequencing. And the right side bar shows the kind of inferred clonality based on the bulk sequence versus the single cell analysis. And as you can see, the single cell sequencing suggested that this triple mutant cells are the most biggest clone, which is a founder clone, followed by this doublet mutant clone. And then the bulk sequencing saw that there is a one big clone with the DNMT3A only, which single cell does not think that there is a big population of that cells. So from this first patient experience, we learned that this platform is feasible in the actual AML patient sample, and we can consistently genotype about 5,000 cells, and right now I think with the updated platform they can go up to the 10,000 cells. And also that there is some potential that this platform can detect this minimum residual disease in the remission samples. And from this patient, particular patient, we were very surprised that actually the diagnosis and relapse sample had almost exactly the same clonal architecture. And I'm going to show you additional two more cases, and we learned that this is not always true, that there is some clonal evolution that happens at the time of relapse. So the second patient was also very similar story, 59-year-old patient with AML M0 who achieved complete remission, but 3 months later, again, relapse. And from the bulk NGS, we knew that this patient had NPM1, IDH2, and DNMT3A mutations. This is analysis of the diagnosis sample and fairly simple clonal architecture of this patient. Big cell population carrying all these mutations, IDH, NPM1, and DNMT3A. And in the relapse sample, this clonal architecture, again, was kind of well preserved. In the relapse sample, almost same exact clonal architecture that this chunk of this triplet mutant exists. So when you compare these baseline and the relapse back to back, you can see that the clonal architecture is almost about the same from the diagnosis and relapse sample, which was similar story like this patient number 1. So the patient number 3 actually had also a little bit unique clinical history because this patient was a 65-year-old guy who were diagnosed with AML with myelodysplastic changes. He achieved a CR also after 1 month, but his CR kind of maintained for about 3 years. And eventually, this patient's leukemia relapsed 3 years later. So the first 2 patients, they relapsed within the 3 months, and this patient had a much longer remission lengths. By the bulk sequencing, we knew that this patient had NRAS, IDH2, and ASXL1 variants. This is a diagnosis samples analysis from this Tapestri platform. IDH2 clone was observed in most of the cells, so suggesting that this is probably the founder clone, followed by NRAS, and small population with this ASXL1 mutation. And this patient's relapse sample that happened after 3 years, there was a significant expansion of this small ASX1 clone that was originally at the diagnosis, now expanded to almost all cells. And also NRAS clone expanded at the same time. They also acquired some of the homozygous state cells. So this was a little showing that different clonal architecture between the diagnosis and the relapse. And as you can see in the right lower area where it shows the reduced dimensional analysis, that there is a shift of clones from diagnosis to the relapse. It could be associated with this patient's this long CR remission compared to other patients who kind of quickly relapse after 3 months. So there may be the clonal evolution associated with the duration of also the remission. But with these 3 patients' experience, what we learned from this platform is that first, Tapestri Platform runs very well to the actual patient's bone marrow sample that were longitudinally acquired. There's a good concordance of both variant detection and as well as a variant allele frequency estimate between the bulk sequencing and also this single cell sequencing platform. And this provides us the very unique opportunity to visualize the precise clonal architecture, not only the combination of a co-occurrence of the mutation in the single cell level, but also with this different zygosity of the mutation. And also, it's suggested that this platform may have some role in the MRD detection if we can analyze much more numbers of cells, and then we can detect the small numbers of clones in the minimal residual disease. I think this single cell platform provides a great research questions and the clinical questions for the future. It adds kind of another layer for the clonal heterogeneity analysis, not only the co-occurrence, but also the zygosity. And how do we interpret this, and how do we incorporate these data into the clonal architecture modeling? This is a new question that we really need to solve after this. And 1 out of 3 patients had a kind of unique clonal evolution pattern, and we really need to learn what kind of patterns are there. So we are very eager and excited to expand this cohort of patients. And all these 3 patients, actually, we selected a case who have a normal chromosomes to avoid this allele kind of heterogeneity. But how do we analyze these chromosomally abnormal cells or copy number, LOH, those stuff? These has to be also researched as a next question. So I'm just going to end, but I'm from Houston, and as you know, when you do the bulk sequencing of Texas, it's all complex to the red state, but we really need to go to the single cell level, because if you look the single cell resolution, actually where I live, which is Houston, is actually blue color, and you really need to understand these heterogeneity in order to win the election and also win the AML battle. So I think I really want to suggest the strengths of the single cell platform. Thank you very much.